Assalamu alaikum. My name is Khadija Abdul Majid, and the topic that I will be speaking on today is the assessment of synergistic antibacterial activity of 10 indigenous plants against locally isolated MRSA, methicillin-resistant Staphylococcus. The need for new phytochemicals arises due to the emergence of antibiotic resistance as well as the shortage of new antimicrobial drugs. Medicine plants can be defined as a group of plants that possess some special properties that make them as potential targets for drugs as well as therapeutic agents. If you look at the use of herbal medicine today, a huge proportion of the rural, undeveloped underprivileged population, particularly in the developing countries, rely solely on the herbal medicine for their healthcare. Phytochemicals are simply chemicals that are produced by the plant through either primary or secondary metabolism. These include alkaloids, flavonoids, polyphenols, anthocyanins, amongst others, and they are responsible for the antimicrobial and other activities that are attributed to the plant. So what is exactly the synergistic action? The synergistic action is one in which the effect of two drugs that are administered simultaneously is greater than the sum of the effects of each drug that is given separately. What is the significance of this study? Staphylococcus aureus is a gram-positive cocci that is found commonly on the human skin, and which is why it is the most common cause of skin and soft tissue-related infections, as well as hospital-acquired invasive infections. Moreover, it also causes staphylococcus food poisoning that is associated with the toxic shock syndrome, pneumonia, and sepsis-related infections. Methicillin-resistant Staphylococcus aureus, MRSA, they are resistant to many antibiotics genetically, and which is why these are a cause of concern for pharmaceuticals. On the other hand, the plant-derived antimicrobials have the advantage of being cost-effective, easily accessible, having lesser side effects, and also being environment-friendly. Moreover, if we take the synergistic effect of these plants along with the antibiotics, the synthetic antibiotics, they may provide fruitful results that would ultimately benefit the community at large. The aims of the present study were to determine the antimicrobial susceptibility of the isolated microorganisms for each of the plants, then to carry out the molecular characterization of the clinical isolate, and then to evaluate the synergistic effect of the crude extract of the plant. Next is the methodology. First, the plant samples were collected from different areas of Lahore. The extracts were prepared by washing them with distilled water, drying them in shade for 15 days, grinding them to a fine powder, which is then used to make the hexane and methanol extracts. Next, the microorganisms with 6 methicillin-resistant Staphylococcus aureus or MRSA isolates were obtained from clinical samples. The next was the molecular characterization of these isolates, where their genomic DNA was isolated. It was qualitatively and quantitatively analyzed, as well as the PCR amplification of the 16S ribosomal RNA gene was carried out to confirm these isolates. To check the antibacterial activity, the agar-well diffusion method was used, and 2 concentrations of the plant mixtures were taken, the STP-100 and STP-15. In STP-100, 10 milligram of each of the extract was taken, and in STP-50 was basically a serial dilution of this concentration. In the results, as you can see from the gel, this gel basically shows the PCR amplification of the 16S ribosomal RNA gene. And as is visible, all the 6 strains had the particular gene showing confirming their origin. Next, the image of the plate basically shows the antimicrobial effect using the agar-well diffusion method. Clear zones of inhibition around the wells show the inhibition of growth in these regions that is inhibited by the plant extract. The graph basically shows the synergistic effect of the 2 concentrations against the MRSA isolates. Your y-axis shows the zones of inhibition in millimeters, and the x-axis has the different strains of the MRSA. And as is visible, the blue bars show the STP-100 high concentration, whereas the red counterparts, they have the severely diluted concentration. And amongst all the extracts, the MDL01 was the most susceptible to the mixture of plant extracts, forming a zone of 22 millimeters, which is quite comparable to that response by vancomycin against this. and so it can be used, it can be possibly potential alternative for vancomycin, having lesser side effects and lower concentration being more similarly effective. So in conclusion, the resistance of antimicrobials is a growing problem, and the future for the use of antimicrobials still looks quite grim. And even though pharmacological industries are producing new antibiotics, a lot of new antibiotics in the last few years, but the resistance to these drugs for microorganisms has also increased, which is an alarming issue. On the other hand, if you look at the plants, naturally available resources of thousands of constituents, which are basically a valuable source of new and biologically active molecules with potentially antibacterial properties. In the present study, we checked the extracts of 10 indigenous plants for the synergistic effect, which they showed against and the anti-methicillin-resistant Staphylococcus, and they found zones of inhibition of diameter that were quite large as compared to that of the individual extracts. And the resulting herbal extracts can be further used to control infections that are caused by multi drug-resistant pathogens, having little to no side effects. Moreover, the antibacterial extracts can also be used in development of biopesticides as per the preservative in processed foods, where they will prevent the growth of microorganisms and also prolong shelf life of food. Thus, they can replace the use of chemical preservatives that have been known to be carcinogenic. Moreover, the antioxidant and antibacterial potential of the plants will also help them to overcome the food security issues by preserving food naturally and also increasing the nutritive and value as well as the availability of food. Thank you.